Good afternoon, children.
Good afternoon, ma'am.
We learned about mutations, two types of mutations we learned, point mutations and frame shift mutations. What is point mutation, Gayatri?
When a single base pair is altered.
When a single base pair is inserted, deleted or changed, you call it as point mutation. Name a disease caused by point mutation, Steffi. I was absent previous period, no? That is what you're going to say. Very good. Anna?
Sickle cell anemia.
Sickle cell anemia. Frame shift mutation, Nandana?
Due to the insertion or deletion of a complete one or two nitrogen bases.
What will happen?
The reading pattern changes.
Reading frame shift. So reading frame will change from the point of insertion or deletion. And now we are going to learn about translation. And about the adapter molecule we discussed. Which is the adapter molecule?
tRNA.
tRNA or transfer RNA. And transfer RNA is having two ends. One is where the amino acid gets attached and the other end is the anticodon arm, where anticodon complementary to the codon present on messenger RNA are present. So let's learn how the process of translation takes place. Hope you are thorough with replication, transcription. Now we are going to learn about translation. You know, as a result of transcription, the information present on DNA will get copied to messenger RNA. Now, this messenger RNA should get converted to a sequence of amino acids, which will result in the formation of a protein. And how that process is taking place, we are going to learn. Translation refers to the polymerization of amino acids to form a polypeptide. You know that polypeptide means? What is polypeptide? Other name? Yes, polypeptide. Amino acids join together and form
Protein.
protein. So protein is a polypeptide, just like DNA is a polynucleotide, RNA is a polynucleotide. Just like that, many amino acids join together. And why we call it as polypeptide rather than polyamino acids? Because these amino acids are linked by peptide bonds. And you call it as a polypeptide. Polymerization of amino acids, several amino acids, the monomer units are amino acids. They are joined together to form a polypeptide or protein. So it is a process of polymerizing amino acids to form a polypeptide. And the number and sequence, how many amino acids or what are the amino acids, in what sequence, which amino acid comes first, what is next, what is the third one, all that is decided by the sequence of bases present in messenger RNA. Clear? Depending upon, you know that each triplet codon codes for a particular amino acid. So depending upon the sequence of nitrogen bases, depending upon the triplet sequences present there, accordingly the amino acids will be joined together to form a protein. The sequence, the order, all that, how many amino acids, all proteins are not having the same number of amino acids. How many amino acids and what all amino acids, in what order, which amino acid will come first and what is next, all that decided by the sequence of nitrogen bases present in messenger RNA. And these amino acids are linked by or joined by peptide bonds. To begin with, for the process to begin, first the amino acids will get activated. Amino acids are activated in presence of ATP. So to begin with, amino acids will be energized. They'll be activated utilizing ATP molecule, adenosine triphosphate. Once they are activated, they will go and link with the cognate transfer RNA. Cognate means related. And how are they related, or how do you know that this amino acid is related to this transfer RNA? Because the transfer RNA is having an anticodon. That anticodon is specific for each amino acid. Each amino acid has got a specific anticodon on the transfer RNA. For each, we have 61 anticodons, 61 transfer RNA. Just like, why 61 anticodons? Because we have 61 codons, and each codon codes for, or each anticodon stands for a particular amino acid. So the first step is charging or activation of amino acids utilizing ATP, and then they will join, they will get attached to their cognate or related transfer RNA. The relation is in terms of the anticodon. So these anticodon is specific for a particular amino acid. This process is known as charging of transfer RNA or amino acylation of transfer RNA. That is, the amino acid is getting activated utilizing ATP molecule, and this activated amino acid will go and bind with the transfer RNA. So that results in charging of transfer RNA or amino acylation of transfer RNA. Amino acid, the charged amino acid is getting attached to transfer RNA. That is known as amino acylation of transfer RNA. So that is the starting step. So here the process begins. Okay, the primary steps. So for the translation process to take place, the transfer RNA should get charged. For that, first the amino acid will get activated in presence of ATP, and they will go and get attached to the ATP. Sorry, they'll go and get attached to their related transfer RNA molecule. So here you can see amino acids. Amino acids, once they are getting activated, they are going and getting attached to the transfer RNA. And here you can see charged or amino acylated transfer RNA. Here you can see the picture of a ribosome and the translation process taking place. When two charged transfer RNAs are brought together, when two charged transfer RNAs are brought together, a peptide bond will be formed between them, and the presence of a catalyst will enhance the rate of this process. So when two charged transfer RNAs are brought together, there will be two charged transfer RNA means they'll be having amino acid at one end. So once two transfer RNAs having these amino acids at one end come closer, a peptide bond will be formed between, suppose this is so this is one transfer RNA and this is another transfer RNA, or let me draw the figure. Just assume that this is our transfer RNA with a particular codon. And then on the other end, there'll be an amino acid. There'll be an amino acid at one end. So when two such amino acids are coming close, when two such acylated or amino acylated or charged transfer RNAs come together, there will be the formation of a peptide bond between both of them. So a peptide bond will be formed between them. To be specific, between the amino acids present. Once again, when two charged means they have got attached with the activated amino acid molecule. That is known as a charged transfer RNA. So when two such transfer RNAs come closer between the amino acids, a peptide bond will be formed. And this will be speeded up in the presence of a catalyst. Have you understood that point, what I said? You want me to repeat again? No, understood.
Yes, ma'am.
You want me to repeat? Yes, ma'am.
Yes, ma'am.
Okay, I'll say once again. I think first part you understood. That is the amino acid. They will be activated. How are they getting activated? In presence of ATP. And once they are activated, they will go and bind with the transfer RNA, their corresponding or their cognate RNA. They will be binding. This is transfer RNA and they will bind with this transfer RNA. And this binding is based on the anticodon present. So these anticodons are specific. So only the matching amino acid will come and get attached. Okay, and this process whereby the amino acid which is activated is coming and attaching to this transfer RNA, you call as aminoacylation or charging of transfer RNA. And then we learned when two such, now you understood that how the transfer RNA is getting charged or aminoacylated. When two such transfer RNAs come closer, when two such aminoacylated transfer RNAs come closer. So this is the anticodon present. And here there will be the amino acid present. Then another charged transfer RNA. Here there is an anticodon. Here another amino acid. So this is the first charged transfer RNA, second charged transfer RNA. When two charged transfer RNAs are brought together, a peptide bond will be formed between both of them. Okay, Nandana?
Yes, ma'am.
And when a catalyst is present, this will speed up the rate of reaction. So that is what is mentioned over there. Now next is ribosome. You know, you had learned in class 11 and even in class 9 about ribosomes. They are the site of protein synthesis. They are the sites of ribosomes are the sites of protein synthesis, or else you can say the cellular factory for protein synthesis. Protein synthesis takes place in the ribosome, and they even act as catalyst. And in bacteria, 23S ribosomal RNA, that is ribozyme, it is present, or ribosome, they can act as a catalyst too, and they act as a site for protein synthesis. So they will catalyze the peptide bond formation and the protein synthesis. You know how proteins are synthesized by joining together of amino acids. When amino acids are joined together by peptide bonds, several amino acids, when they are joined together by peptide bonds, that will form a polypeptide, and this polypeptide is known as a protein. And this synthesis, the process of synthesis of proteins is taking place in the ribosome. Ribosome is a site of protein synthesis, or a cellular factory in the cell. It's a cellular cell organelle where synthesis of proteins takes place. And it also acts as a catalyst to facilitate the peptide bond formation. 23S. Children, this S indicates the sedimentation coefficient or Swedberg coefficient, the rate at which it gets sedimented. You have learned in class 11. No need to worry about that. It is a sedimentation coefficient. 23S ribosomal RNA. In bacteria, it acts as an enzyme and you call it as ribozyme, and it will promote peptide bond formation. And ribosome is having ribosomal RNA and so many proteins. So 80 different proteins and ribosomal RNA are present in a ribosome. What is the significance of ribosome? It is a site of protein synthesis. It is in the ribosome that new and new proteins are being synthesized. It already contains 80 different proteins and ribosomal RNA. This ribosomal RNA can act as enzyme or catalyst. In inactive state, ribosome 70S will be present as two subunits. A large subunit known as 50S, a small subunit 30S. So there will be two subunits for a ribosome. So like this, they have two subunits. Okay, and only when they are active or when they have to synthesize proteins, these two subunits will join together. Otherwise, they will remain separate. So these are the subunits of 70S ribosome. Now, you might be thinking, why 50S and 30S together is making up 70S ribosome? I think you know that. Last year, we had learned 50S and 30S. The sedimentation coefficient of large subunit is 50S and smaller subunit is 30S. But together, they sediment at a lesser rate. So, it is 70S. Together, they sediment at 70S. Individually, they sediment at 50S and 30S. S stands for Swedberg coefficient or the rate at which it gets sedimented. And 50S is the sedimentation coefficient for large subunit, and for the small subunit, it is 30S. But together, when both are together, their sedimentation rate will change and it is just 70S. That is why we call it as 70S ribosome. Any doubt in this? We learned about ribosome. Ribosome is where we are discussing about translation. In translation, you know that protein synthesis is taking place. Amino acids are polymerized to form a polypeptide. Then you should know where it is taking place, the location of protein synthesis, and the catalyst which helps in speeding up the process. So ribosome is the answer for that. It is in the ribosome that the protein synthesis takes place. They also act as a catalyst to facilitate peptide bond formation between the amino acids. Okay, children?
Yes, ma'am.
Okay. Now here you can see the translational unit. Translational unit in a messenger RNA is a region flanked by start codon and stop codon. Flanked means bordered by one end you can see start codon, other end you can see stop codon. In the figure, see the figure here, this is a translational unit. There is a start codon at one end and stop codon at one end. So this is known as a translational unit. It is flanked or it is edged or bordered on either side by start codon and stop codon. So translational unit in a messenger RNA means it's a region which is flanked by start codon at one end and stop codon on the other. And then see the next figure. Here you can see start codon. Start codon is here. This is start codon. Here it is stop codon. But you can see on the 5' end yellow colored part and on the 3' end some purple colored part. And those are not included under the translational unit. You call them as UTR or untranslated regions. These are regions on messenger RNA. They don't take part in translation, but they are necessary for the translation process to proceed. For translation to take place, these UTR, untranslated regions, are needed. They are not getting translated into proteins, but their presence is needed for the translation process to take place. And where are they present? They are present at the 5' end and at the 3' end. At the 5' end before the start codon, and they are present at the 5' end before the start codon and at the 3' end after the stop codon. Once again, translational unit. Translational unit means that is a region of messenger RNA which is bordered on one side by start codon and other side by stop codon. On 5' end start codon, 3' end stop codon. And before start codon and after stop codon, you can see some untranslated regions. They don't take part in translation, but their presence is needed for the translation process to take place. And as I told you, they are present before the start codon at the 5' end and after stop codon at the 3' end. Now we are going to learn about the different steps involved in translation process. To begin with, initiation. So before I start, I will just explain a few points. I think these are not mentioned in your textbook. Children, this is the small ribosomal unit. What you see here, this is messenger RNA. And this is amino acylated or charged transfer RNA with a amino acid attached at one end and anticodon on the other end. That is the transfer RNA. And this is a small subunit. And here you can see a large subunit. And the large subunit of the ribosome has got three sites, E, P, and A site. The large subunit of ribosome is having three regions, E, P, A. E stands for exit site, P for peptidyl site, A for acceptor site, amino acyl acceptor site. So it is where the amino acid is getting accepted. Three sites are there, not in the small subunit, but in the large subunit. Three specific sites are there. EPA site, E exit site, P peptidyl site, and A acceptor site or amino acyl acceptor site. These are the three sites. Now for the process to begin, initiation. The process of translation or protein synthesis begins with the attachment of messenger RNA with the small unit of ribosome. So when the process is beginning, the ribosomes, both the subunits are not there. To begin with, the messenger RNA will bind with the small subunit. And the ribosome will be attaching to the messenger RNA where at the start codon. The start codon is AUG. AUG codes for methionine and also it is a start codon where the process begins. Okay, the process of translation begins when the messenger RNA will bind with messenger RNA binds with the small unit of ribosome. And where the ribosome is getting attached, it will get attached at the start codon or AUG. Now this AUG is the codon, start codon as well as it codes for methionine. So once this ribosome is attached to the messenger RNA, the start codon, this AUG will be recognized by the initiated transfer RNA. Initiated transfer RNA is the RNA having anticodon for AUG. See, anticodon will be UAC and it will be having methionine at one end. Okay. And this transfer RNA will come and get attached to the start codon because there it is AUG. So that is how the process starts. Once again, to begin with, the messenger RNA, which has to get translated, will get associated with the small subunit of ribosome. Now, where it is getting attached at the start codon. Now, the transfer RNA having the anticodon UAC, they will recognize this AUG and they will come and bind there. Here you can see it is having methionine. See, first I'll explain with the help of this figure. This is the, where to begin? Okay, here. So here I told you EPA site. EPA site, and here in the P site, it is at the P site that the initiator codon is present. The transfer RNA amino acid complexes bind to their corresponding codon on the messenger RNA, and complementary base pairing takes place between the codon on messenger RNA and transfer RNA anticodon. Children, in the previous slide, I told you the process begins when messenger RNA gets attached with a small subunit of ribosome, and the initiator transfer RNA. Initiator transfer RNA means the transfer RNA having anticodon for AUG, or which is having methionine, or the transfer RNA with the amino acid methionine is known as the initiator transfer RNA. They will come and get attached. They will come and get attached over here. Once this has happened, this bigger unit will come and get attached. Once this process is completed, that is, initiator transfer RNA has got attached to the corresponding codon, that is AUG, on messenger RNA, then the large subunit will come and join the smaller subunit, and then you can see it is in the P site that the initiator transfer RNA is present. E and A sites are free. Okay, once the large subunit come and get attached to the small subunit, the positioning is like this. This P site, this E, P, and A sites are present only on the large subunit. So when they are coming and getting attached to the small subunit, the initiator transfer RNA will come at the P site. Now I'll show you the next figure. Not to worry, children, if you're not following. I'll show you the video also. And then what happens is then another messenger RNA, another transfer RNA. So this is, just for example, you assume that this is methionine. This is methionine. Okay. And then at the A side, I told A is the amino acyl acceptor site. So here another transfer RNA will come, which is having anticodon to the codon present on the A site. So they will come and fix at that position. So this is having one amino acid. This also will be having another amino acid. Now, once these two has come closer, I told there will be already I told you there will be a peptide bond formation between these two. So once the peptide bond has formed, this amino acid will get separated from this initiated transfer RNA. It will be transferred to the transfer RNA present at the A site. So the one which is present at the P site is free now. Then this ribosome will move so that the positions will change. The one which is present at the A site will come to the P site, and the one which was present at the P site will reach E, and from there it will be leaving outside. That is the exit site. So here at the P site you have transfer RNA with 2 amino acids, and again at the A site a new transfer RNA carrying amino acid will come, and this process will get repeated. I don't know whether you have followed it. Before I proceed further, I will show you the video. I think after showing the video, if I explain, you will understand it better. Just a minute, children. I'll show you the video. That is easy for you to understand. Okay, now let's see it once again. So from the video, you might have got a better idea. Initiation, the process of translation or protein synthesis begins. As you already saw, messenger RNA will come and get attached with the small subunit of ribosome, and then the AUG. So ribosome is getting attached at the start codon because the process is beginning there. And the ribosome, as the process proceeds, it will move from the start codon to the stop codon of the messenger RNA. So to begin with, messenger RNA or the small unit of ribosome will come and get attached to the start codon of the messenger RNA. And this will be followed by the recognition of AUG. AUG is the start codon where the translation begins. So initiated transfer RNA. Initiated transfer RNA is the one which is having anticodon for methionine. And you know that start codon, it codes for methionine and it also helps in beginning the process of translation. So AUG will be recognized. AUG is the start codon, will be recognized by the initiated transfer RNA. And this initiated transfer RNA is having methionine. And then once this has taken place, next step is the large subunit will come and join with the small subunit. After the transfer RNA, initiated transfer RNA has attached to the AUG or the start codon, next step is the large subunit will come and join it, and together they will form translation initiation complex. That is not mentioned in the textbook. If it is not mentioned also, no problem. So the large subunit will come and join together. And I told ribosome is having 3 specific sites. E, exit site. It is from there that the amino acid, it is from there that the transfer RNA, after transferring the amino acids, it is being released from the exit site. Then P site, it is at the peptidyl site that the process begins, and at the A site, it is from the A site that, or through the A site that new and new transfer RNA molecules are entering the ribosome. So to begin with, I told it is occupying the P site. The transfer RNA, initiated transfer RNA is getting attached to the P site. Now, once it has got attached, now next transfer RNA, another transfer RNA will be entering into the A site. That also will be, see, another amino acid will enter here. So P site, this is initiator transfer RNA with methionine. Through the acceptor site, another one will enter. Here another amino acid will be there. And then what happens? A peptide bond will be formed between these amino acids. Once the peptide bond is formed, this will, or the transfer RNA present at the P site will transfer the amino acid to the one which is present at the A site. And then the ribosome will be moving along the transfer RNA. So the positions will change. The one which is present at the A site will reach the P site. The one present at the P site will reach the E site. And at the E site, this is not having any amino acid. So that will be leaving from the E site. Now you can see 2 amino acids here. Again, the process continues. Another transfer RNA with amino acid will come here. It will be having an amino acid. Then what happens? These 2, there will be a peptide bond formation, and these 3 will be transferred here. This will move to this side, and from there it will get exit. And from the A side, it will move to the P side, and the process keeps on getting repeated. Transfer RNA amino acid complexes bind to their corresponding codon. Children, what you need to remember is all the new transfer RNAs entering through the amino acyl acceptor site, they are having the corresponding anticodon which can fix with the codon on the messenger RNA. If this is not matching, they cannot bind over here. It should be matching. The anticodon present on the transfer RNA should be matching with the codon on the messenger RNA. Only then they can fix and the process can continue. The ribosome moves from codon to codon. That is what I said. This is the messenger RNA, and the ribosome will be moving from one end to the other. So as the process proceeds, the messenger RNA, along the messenger RNA, ribosome will be moving from codon to codon. They are then translated into polypeptide sequences under the control or dictated by DNA and represented by messenger RNA. Under the directions of DNA and by translating messenger RNA, polypeptide sequences will be formed. And next is termination. So elongation, you might have understood. So what we were discussing, this was about elongation. Initiation, we learn how the process starts and how the polypeptide chain is getting elongated, we learn. And then the final step is termination. So termination takes place when the ribosome reaches the stop codon. So a release factor also will bind to the stop codon and will facilitate the process to come to an end. And once this process is completed, the polypeptide, the formed polypeptide will be released. So polypeptide is released, messenger RNA is released, the two subunits are getting separated, and the release factor, everything will get dissociated, and again they will associate for another round of translation. See again one more symbolic figure, a symbolic video. Just see that. See, children, why it is shown as one amino acid is joining and again it went. No, it was shown like that because it's not having matching anticodon. That is why it was rejected. And see, the ribosome is moving along messenger RNA. Then another one again enters the A site. And this process will continue till the end or till the stop codon. See again, methionine coming and getting associated to AUG. Small subunit is getting associated. Then the bigger subunit joins. See, an amino acid GUG anticodon comes, but that is not matching, so rejected. Another one having matching anticodon will come. And now you can see a peptide bond is formed between those two amino acids. Again, another one joins. Peptide bond between them. And this process will be continued. And after transferring the amino acid, the other transfer RNA will be released. Have you understood? Somewhat? Majority of the videos are off. I'll send you the video and just go through that. And once again, I will repeat the process. You see it. I don't know whether it was getting stuck in between. I don't know. So I will send it and see it, observe it carefully. And once again, I will explain the translation process. Okay, after seeing, all should see the video before joining the next class. So that both transcription and translation, I will send EPA sites. About that, it's not mentioned in your textbook. But if you need to understand the concept clearly, I think you should know about EPA sites. E is the exit site. And from the exit site, then non-charge or after, when is the transfer RNA getting charged? When it is having that amino acid. But once it transfers this amino acid to the other transfer RNA, its charge is going. So once it is not having any charge, it will be released from the exit site. And through the A site, new transfer RNA will enter. Then the one which was present at the A site, P site will move to E site. The one which was present at the A site will move to the P site. The process gets repeated. I don't know how far you have understood. How much did you understand, children? Little more clarity is needed?
Yes, ma'am.
Yes. So you see the video, translation process, I'll repeat again in the next class. So if you haven't followed, not to worry, I'll be repeating it again in the next class. But you should see the video. Only then, once I'm explaining, because in your textbook, many points are not there. If you read the textbook, the whole process you can't understand clearly. But if you see that video and then you read the textbook, it will be easy. So we can have it once again in the next class. Okay. So I'm not asking you any questions related to translation. After the next class, once you have understood the concept thoroughly, I'll be asking.